Assalamualaikum warahmatullahi wabarakatuh. We are from the last group and today we want to present about biological control papers implementation of predator as biological agent. Let me introduce you our team member. The first one is Sara Vanesya with student number 131. Rinta Sofia with student number 145. Safira Tasya with student number 171. And Alifiandra Soni Putra with student number 208. The first one is predator characteristic. The predator eats a number of prey during its growth and development process until it reaches adulthood, except for egg or cocoon stage. All life stages for predators are free-living. Predator eggs are usually laid near colonies of their prey. Once hatched from the egg, the predator nymph or larva actively seeks, catches, and kills and eats its prey. Many carnivorous predators are either nymph or larva or adult stage, except for a few which are not egg-earfit flies. The next one is predator eating behavior based on the eating mechanism. A predator that has chewing mood tool eats and swallows its prey. For example, dung beetles, carpet beetles, praying mantis. And the second one is predator who has a sucker stick their stingray into the prey's body and then suck their blood out. These predators often have strong venom and digestive enzymes that cause their prey to be paralyzed. For example, pentatomid ladybugs, mayfly larvae, and stirfid larvae.
Predatory eating behavior based on the host range. First, we have monopagus, a type of predator that severely limits the range of its host, sometimes limited to one type of host, example, Vedalia beetle. Second, oligopagus, a type of predator that somewhat limits the range of its host, limited to a few types of hosts, example, apid-eating and sulfidome beetles. Third, polyphagus, a type of predator whose host range is wide, sometimes limited to one type of host, example, green net flies and praying mantises. This is the Protura, Embioptera, Zoroptera, Isoptera, Malovaga, Anoplura, Homoptera, and Siponaptera. Araneae and Acari predators. Research on the effectiveness of the spider as a predator is still limited. However, many studies show that spiders are not adapted to the fluctuation in the population of a pest species. Spiders are considered as stabilizer of the invertebrate community because they move from one type of prey to another when there are many other types of prey. Most attention has been paid to pitosaid mites as predators of tetranicid mites. Due to their relatively small size, predatory mites can live in very low prey population. Examples of pitosaid predatory mites are Pitosaurus persimilis and Ambiseus fallacis.
The next one is biological aspect of predatory insects. Living stadium, egg, placed by the parent in groups or singly. Name or larvae body parts are modified when compared to phytophage insects. The position of the mouth becomes horizontal, the forelegs become fractorial to make it easier to catch prey. The intestines are longer and the salivary glands produce alkaline secretion to immobilize prey when eating them. In some cases, the mouth apparatus of the nymph or predatory larva develops into an effective tool for sucking the prey's blood. Imago sometimes have the same type of prey as the pre-adult stage, but also others such as pollen. The next one, predatory relationship with prey. Searchability, the predator responds to environmental focus to its prey, habitat selection, prey discovery, prey acceptance, and prey sustainability. Most imago predator require a minimum number of prey to lay eggs and lay eggs. A minimum number of prey must be eaten by the pre-adult stage of the predators to meet energy requirement for survival. Foraging for prey, growing and developing. The number of prey required by a predator depends on the size of predator's body, length of time for prey and other activity, the size of the prey and its nutritional quality. The ease with which a predator can find prey depends on searching efficiency by predators, prey population level, the spatial distribution of the prey, and difficulties or obstacles experienced by predator in the prey habitat. The next one, spawn and predator stage. Eggs are laid near the prey. Only pre-adult stage are predators. For example, Diptera, Hymenoptera. Second one, pre-adult and imago as predators with the same type of prey. For example, Neuroptera, Thysanoptera, Coleoptera, Coccinellidae, Coccid predators, Coccidulanae, Chilocorinae, Sticotolinae, and pre-adult and imago stages as predators with different types of prey, for example, Diptera. Eggs are only laid in the general environment of the prey. Only pre-adult stages are predators, for example, Diptera, Tabanidae, and Bombylidae, usually aquatic like Lecoptera, Trichoptera, some Neuroptera, and Odonata. The third one, eggs are laid freely regardless of the position of the prey. Only the pre-adult stage is predatory. For example, Meloidae, pre-adulthood and image stage as predators with the same type of prey. For example, Orthoptera, Thysanura, some Hemiptera, pre-adult and imago stage as predator with different types of prey. For example, Manticipadae, Raphidoidea, only the imago stage is predatory. For example, Mecoptera, Diptera, and Hymenoptera.
Okay, the next one is predator impact. Classic biological control. Only about 11% of biological control programs using predators are successful. A number of pest species have been controlled by several types of predators. The type of pests that have been successfully controlled by predators are those that are relatively passive, do not use and do not migrate, such as shield lice, leafhoppers, and pests. Attributes of successful imported predator include multivoltines, no diapause, monophagus or oligophagous, high efficiency of search power with a long lifespan, and in accordance with the habitat condition of the prey. Examples, Vedalia beetle. For the control of the Aiseria orange, we will purchase for muscle, Cryptognatha nodiceps for the control of coconut shield lice, the dome beetle Curinus coeruleus mulsan for controlling the Lamtoro heterosophila cubana. Reasons for the failure to introduce predators include the climate where the predator was released is not similar to the original area of origin, lack of prey, and absence of symbiotic microorganisms. The impact of predator on agricultural habitats is difficult to demonstrate because it is difficult to isolate the influence of these predators from other natural controlling factors in field conditions. Dubach argues that predatory polyfags are a counterweight to the natural enemy complex pests. However, perfect natural control was never attained. These generalist predators only inhibit the rate of increase in the population of many species. In the cotton ecosystem, this predator like Geocoris, Orius, Napis, and Chrysopa often prevent the outbreak of pests such as armyworms and jengkal caterpillars. Predator augmentation. Additional feed is distributed in the crop to arrest predator or to keep predators retained or attract predators to increase their laying on the land. This additional feed is in the form of artificial honeydew, which is a mixture of sucrose, yeast, and sugar. Pollen can also be spread in the fields for augmentation of predatory mites. Other augmentation efforts can be done by releasing predator in an inundative way. For example, Cryptoleamus montroseri, bug destroyer. The predatory mite Phytoceulus persimilis. Okay, conclusion is predator that can be used as biological agent comes from Ordo Protura, Ambioptera, Zooptera, Isoptera, Maluvaga, Anoptura, Homoptera, Siphonaptera. There are two types of predatory eating behavior based on the eating mechanisms and based on the host range. Biological aspect of predatory insect are living stadium, predatory relationship with prey, and spawn and predator stage. There are three predator impact: classic biological control, the original predators of the agro ecosystem, and predator augmentation. Okay, that's our presentation. Thank you.